Aletta Millen and Sooraj Baijnath started the Integrated Molecular Physiology Research Initiative about 2 years ago. Their lab focuses on the understanding of the molecular mechanisms underlying several NCDs using advanced molecular techniques. One of the techniques they use, and the focus of today's seminar, is mass spectrometry imaging. The initiative currently hosts the first atmospheric pressure matrix-assisted laser desorption, laser ionization, mass spectrometry imaging system in Africa. That should really be done to music, I think. It just has a certain rhythm to it, which they want to use to solve specific African healthcare problems which may not be investigated in the global north and very well would not be prioritized. Aletta and Sooraj are privileged to collaborate with one of the world leaders in mass spectrometry imaging, and Sooraj spent 2 years in Per's lab as a researcher. So Professor Per Andren is a professor of mass spectrometry imaging at the Department of Pharmaceutical Biosciences in Uppsala University in Sweden. Professor Andren is interested in the use of MSI, mass spectrometry imaging, for the analysis of biological systems and focus on MSI method developments and applications of the brain and neurodegenerative diseases, with particular emphasis on L-DOPA-induced dyskinesia in Parkinson's disease. His laboratory is a Swedish national facility for MSI within the Science for Life Laboratory, otherwise known as SciLife Lab, and an institution for the advancement of molecular biosciences in Sweden. So we're very pleased that you're here with us, Per, and very happy that Aletta and Sooraj can open this and provide, I think, some awareness of what this new technology can bring to this group, and of course we're also connecting them to an array of scientists throughout IDORI, because these capacities are truly amazing, as you'll see. So I think I'll turn this over now to Aletta. Thank you very much.
Good afternoon, everybody, and thank you very much for coming. So we are very excited to share with you a little bit today, and we decided to break it up into the three of us so that you don't get too bored to listening to just the one of us the whole time. So hopefully it keeps you awake. So as Professor Bryman mentioned, so about 2 years ago we started the Integrated Molecular Physiology Research Initiative, and a large part of that is centered around using mass spec imaging in order to advance several questions and healthcare issues which plague South Africa, and we believe that really this technology is vital in improving our understanding of health and disease. So we want to show you today, it's a bit of a tech talk, so I apologize for that, but we want to show you how we can use this technology and really apply it, and how we can advance many research questions in our faculty. Now, you're well aware of that most molecular studies have really advanced our knowledge of health and disease. But current molecular and biological workflows usually depends on homogenizing the tissue. So whether you want to do transcriptomics or metabolomics with mass spec, or whether you want to do proteomics with Western blots or ELISAs, you need to homogenize your tissues. And basically, you are losing all the spatial information of the tissue. So I want to first establish why is it important for us to actually look at spatial information. Why can't we just homogenize the tissue? So you are very well aware that most organ systems consist of several different cells, and each of the different cells have a different function. And also, when you look at drugs that target several diseases, where the drug goes in cancers or in neurological disorders is really important for us to characterize in order to see whether the drug actually works optimally. So the first example I want to talk about a little bit is neurodegenerative diseases. So it is well known that several neurodegenerative disorders, including depression, Parkinson's disease, Alzheimer's disease, is only located really in certain areas of the brain. So if we now have an animal model and we homogenize the entire brain, we lose the spatial information of those animal models. Similar in human diseases, the same can be said. Another interesting and very important reason why we need to study spatial information, and specifically for IDORI, is that we know that most cancers are really very heterogeneous. There are several different cell types, and each of these cell types basically perform a different function. And the different parts of the cancer is also very different. So understanding exactly how the transcriptomic, metabolomic changes in the different parts of the cancers happen really will provide a huge advantage for us in order to be able to design drugs which could target the very specific cell types, very specific functions of these various cancers. So this is just two of the reasons why spatial information is important, and I'm sure you can apply it very well to each of your own research areas. So I'm pretty sure I've made my point that spatial information for us is really important. Now, although there are various spatial omics techniques available around the world, so there's spatial transcriptomics, spatial metabolomics, and spatial proteomics, today we're going to focus mostly on the metabolomic aspect of it, because we want to focus on the technique of mass spectrometry imaging and how that can benefit what it is that we want to look in terms of this spatial information. So if you look at this slide, when I put together the slides last week, I just Googled the number of papers that have been published with the word mass spectrometry imaging in it. And already by the 9th of November, which is about a week ago, there were already more than 1,200 papers this year published with this technology. But you will see how the technology has developed over the years. And really, in 1997 was the first time that mass spectrometry imaging was used in order to understand biological applications. Prior to that, it was used in physical chemistry and using to image semiconductors and stuff like that. But only in 97, really, that they start using it for biological applications. And the very first paper that came out of mass spectrometry imaging in a biological sample was published in Analytical Chemistry. And it was developed by this guy, which name is Richard Caprioli. He's really the father of mass spectrometry imaging. And at the time, Per was actually a postdoc in Richard Caprioli's lab. So Per has really he's developed and matured with the technique over the years. So we are really privileged to work with him. Subsequent to the technology being used for biological applications, really in 2001, 2020, that was the technique highlighted in Nature Methods as the method of the year. And subsequent to that, you can see the exponential rise in the number of publications with mass spec imaging. And you will also see that there are several pharma companies who have adopted the technology, and also most of the techniques, or most of the manuscripts and studies that use mass spec imaging, the studies are published in really high impact journals. So Rob said that we need to put music to this, and I do agree. So it's a huge word. So what we currently have in our lab is what we call a matrix-assisted laser desorption ionization mass spectrometry imaging system. Okay, so this word is ginormous, or the name is ginormous, and I'm going to try and unpack it by showing you the workflow and how this technology actually is used. So the first thing you need to do is obviously have your biological sample, and one of the values of mass spectrometry imaging is that you can use basically any biological sample. You can use any biological fluid, cerebrospinal fluid, blood, plasma, and even interstitial fluid, and or any tissue section, a tumor biopsy, or a whole brain from an animal that you can section. So the best preparation of tissue is actually fresh frozen tissues, but you can also use the technique on FFPE preserved tissue. So once you then have your tissue sample and your tissue preparation, you section the sample using a cryostat, and then you place your sample on a glass slide. And this is where the matrix assisted comes into the word. Because once you have your tissue section, you will then apply a matrix with a robotic sprayer over your tissue section. Now, there are several matrices which have been developed, and that is also one of the things that PAR is heavily involved in, is the development of matrices to ionize very specific and difficult to view compounds. So depending on the matrix that you use, certain matrices are more geared for certain types of molecules. So there's ones geared for specifically for lipids, some for peptides, some for small molecules, and some for drugs. But what is very important is that you can use the same tissue section and apply a matrix, image your peptides, then you wash off the matrix, apply a different matrix, and then you can image lipids. So in the same tissue section, you can use different matrices and then view various different compounds. So then the next step, obviously, is the data acquisition. So I'm going to spend a little bit of time on that to show you exactly how does the technology work. So once you put your slide into your mass spectrometer, the laser then rasters across the tissue. And as the laser rasters across the tissue, it generates a mass spectrum at every laser point. So as the laser hits the tissue section, the matrix allows whatever is in the tissue to be ionized, and that plume of ions is then entered into the mass spec, and each of these ions then is generated as a single biological molecule in your mass spectrum. So what you then end up with is basically, so your tissue section and each of the laser spots basically then generate a pixel. So each of the laser spots is like taking a small picture of that part of the tissue. And each of these pixels then is bound to, or it's connected to, a mass spectrum. So in every single tissue spot, you can then generate a mass spectrum. And when you overlay these, this information, you can find the distribution of several different compounds in that tissue section. So this is just an example of the things that you could measure. And you will see that the intensity of, or the abundance of the compound that you measure is usually indexed by a higher color, so more towards the red side, and if the compound is absent, it would be blue or black. So, as I mentioned, mass spectrometry imaging can measure both endogenous as well as exogenous compounds. So you can measure drug distribution in your tissue section, or you can measure several different kinds of metabolites, neurotransmitters, lipids, and neuropeptides, or whatever it is that you are interested in. So there are several advantages of mass spectrometry imaging. So although it is very expensive to obtain the equipment, as we have been battling for the last 2 years, but once you've acquired the actual instrument, running the experiments is actually very cost effective, because it doesn't require antibody labeling, and you can basically use the same tissue section over and over again, depending on what it is that you want to measure. Now, some of the advantage of mass spectrometry, I'm sorry for lots of wording, but it's got a very high sensitivity and specificity. You can also use several different kinds of techniques on the same image. So you can do multimodal imaging. When you put your image in, or when you put your tissue section or through the laser, it doesn't destroy the tissue, so you can use your same section again, and you can quantify the analysis of the compounds that you are looking at. So, as we know, here in the armpit of Africa, money is hard to come by. So we have, through frugal innovation and having to make a plan, coupled a mass spectrometer and a third-party imaging source to make what we currently have in our lab. Through a partnership with Anatech, we have then also obtained a robotic sprayer in order to use the mass spectrometry imaging. So there are several applications of MSI, which is what we really want to show today, and Sooraj is going to take over from here. But the one thing I just want to end off with is that since we have obtained the instrument about a year ago, there's actually been quite a large uptake in faculty from researchers who see the value and of the technology and what we can actually do with this. So Sooraj will show you a little bit about what we've done so far in our lab and how we have done some experiments with other researchers in the faculty.
Thank you. Okay, so thank you everyone. Thank you for joining us today, and we are really excited about this technology and really excited to be driving this in Africa to kind of show the applications. And like Aletta said, one of our most important goals is to show how this technology can be used in African problems. So I'm going to give you just a few examples of what we've been using the technology for and some of the data that we've been able to generate, which I'll say, although we use some frugal innovation, I would like to think that our images are quite beautiful. So if you think about when mass spectrometry imaging first started, it started off with people mostly focusing on drug distribution and trying to understand the distribution of different drugs in tissue sections. And I guess that's quite an easy application because it's easy for us to buy a standard of a drug, put it on a MALDI target, look at the mass of the drug, and then compare it to what we find in tissue. So some of the early applications that we had was looking at the distribution of HIV drugs in a rodent brain. So we administered these drugs to these animals, and over time, we terminated them at different time points. So at 0 minutes, 15 minutes, half an hour, 2 hours, and 4 hours. And what's really interesting about mass spectrometry imaging is that it allows you to track the entry of the drug into the brain, look at its distribution in different compartments, and look at how the drug is excreted or eliminated over time. I think this is the real value because when we do pharmacokinetic analysis, we're able to measure the concentration of these drugs and the excretion of these drugs. But it doesn't really give us much information about where the drug actually localizes in tissue. And that's kind of where this started off and why I like to focus on the brain. And maybe this is why Per also likes to focus on the brain, because the brain has all of these really cool compartments, and it's really nice to see that the drug gives you these really cool features in the brain. You wouldn't think of the brain as kind of having all of these. And the reason why this came about was that some of you may know that we have a problem with HIV-associated neurocognitive disorders and HIV-associated dementia, and we have TB meningitis. So we know from MRI and PET scans that specific regions of the brain undergo neurodegeneration and atrophy. So we wanted to try and understand, like, firstly, can these drugs cross the blood-brain barrier? Are they able to localize in these regions where HIV causes these types of dysfunction? So the next one, so this one's also, like Aletta said, we try to include some examples that are relevant to IDORI. So this was quite an interesting example that I found. So this is a cancer tumor that was treated with an anti-cancer drug, paclitaxel. What was super interesting is that you see that this drug has a preferential distribution, and when you correlate it to H&E stains, you find that the drug actually doesn't penetrate to the necrotic core of the cancer. So this is kind of the value of mass spectrometry imaging. It allows you to evaluate a wide range of drugs and determine, is it able to get to where you really want it to get? Because I guess in a cancer, this is where the real damage is taking place. But if the drug is not able to reach that location, it really can't carry out its function. So Per will know, and this was a challenge that we faced a lot during the early days of mass spectrometry imaging, is that most publications wanted you, especially if you did drugs, they wanted you to have an image of the drug distribution, and they also wanted you to include some LCMS data. Because at the time, and even now, LCMS is the gold standard for quantifying drugs and looking at drug penetration and drug distribution. But mass spectrometry is not, because you get this heat map, and the heat map or color scale gives you an idea of the abundance of the drug. So Per had a PhD student in his lab, Patrick Shellback, who then decided to compare LCMS to mass spectrometry imaging. So what he did was he took a rat brain that was treated with citalopram, he took serial sections for MALDI imaging, the next set of sections for LCMS, and then again MALDI imaging with a different instrument. And what Patrick then did was he compared, or he followed the FDA and EMA guidelines for developing a bioanalytical method to test the linearity and the reproducibility, and then he also carried out the same experiments on tissue sections to see if mass spectrometry imaging was truly quantitative and if it can be compared to LCMS. And what he found was that there was actually very little variance between the LCMS data and the MALDI MSI data, with the added advantage is that with the MALDI MSI data, you also get a heat map which shows you the distribution of the drug in the tissue as well as in concentration. So then we look at some other kind of, and we spoke about this earlier today. Over time, the technique has kind of matured. Initially, lots of time was spent in developing different types of instrumentation, sample preparation methods, matrices, to the point now where the technique has really matured and it's available for going out into the clinic and having clinical applications. So most mass spectrometry imaging scientists, they love to image lipids because lipids are abundant in all biological tissue and ionize really well. And I think the value in lipids is that lipids allow us to look at the structural components of cells. And in this example that I found, we can see that there's 12 lipids on this image, and these are lipids that were imaged in the small intestine. And what's really interesting is that it allows us to look at the individual villi in the intestine. So by imaging lipids, we can look at structural alterations in the cell and then correlate them with H&E images. So aside from giving us information about exogenous compounds, we can also study structural changes in tissue using mass spectrometry imaging. Okay, so this is also another cancer example. So this is a cancer tumor which shows the distribution of different peptides in the cancer tumor. And we can see, looking at the H&E stain, that these peptides localize in very specific tissue compartments. And we'll show you an example later that we did in collaboration with Mandeep's group, where we see these changes also in our own data. We see that there's very specific regional changes in the biomolecules within these cancer tumors, and maybe that can point us into what's actually going on in the biology of these cancers. So this is an example from Paz lab, and we'll show you a bit later. So traditionally, when people think of mass spectrometry imaging, we think that we are limited to tissue samples. But what's really interesting and what's worth exploring is that mass spectrometry imaging can be conducted on any biological tissue, blood sample, CSF, interstitial fluid. And the way that we see it and the way that we imagine it is that there's lots of potential for it to become a rapid diagnostic test, because it doesn't take us very long to image a tiny spot or 1 microliter spot of the biological fluid. And this example that I will talk a bit more later was CSF samples from Parkinson's disease patients. So they were able to image these spots, and when you do a PCA analysis, you can see that the Parkinson's disease and the control patients actually split up very easily. So using the data generated from these spots, you can actually characterize them into disease and healthy patients. So this again is something that Per developed in his lab, and his lab has become world-renowned for, is the imaging of neurotransmitters. So typically, neurotransmitters and small molecules are really difficult for us to ionize because they're low molecular weight. They fall within the mass range of the matrix, so it's very difficult for us to differentiate them from the matrix. So what Reza from Paz lab did is that he developed this reactive matrix. So this is something that we can do, and another avenue for research in mass spectrometry imaging is developing reactive matrices that are able to bind to specific functional groups on compounds that allow us to help them ionize better and derivatize them. And using this, we were able to see that if you take 6-hydroxydopamine lesion rat, where we destroy the dopamine neurons on one side of the brain, we can actually image the dopamine changes and image the absence of dopamine in the side of the brain that was lesion and that has dopamine neurodegeneration. So this was something that we did last year from our lab. So this is quite a focus of our lab in studying the role of inflammation in developing cardiovascular and neurodegenerative disorders. So we then applied the same technique to animals that had systemic inflammation to study kind of what are the neurotransmitter changes taking place in the brain. And what we can see is that systemic inflammation really drives decreases in monoamines in the brain, which is typical of depressive-like phenotype, which is what we were trying to replicate. And this is an example also from Paz lab. These are post-mortem brains from human Parkinson's disease patients, and you can very clearly see that there are specific neurotransmitter alterations in the brains of these patients who have Parkinson's disease. So this is some data from literature and some data from our lab. So the data that I show you now are data that we collected on the instrument that Aletta showed you earlier. So the first example is from Mandeep's lab. So Mandeep gave us a colorectal cancerous tissue that we thought we'd do some trial runs to kind of see what's going on and if imaging is actually applicable to this tissue. So when we did the image and we look at the total ion current, so like an overview image of the tissue section, we see that there's really this compartmentalization in the tissue. So then we try to understand what's going on, and is this something real or is this an artifact in the sample? So then we gave the tissue back to Mandeep's lab, and we said, like, we see something interesting going on. Could you do an H&E stain on this? So when we did an H&E stain, we see that this compartmentalization is actually the stroma of the cancer versus the tissue surrounding the cancer. So then we went on to look at the distribution of different compounds in this cancer tissue. And what was really interesting is that we found specific lipids that are associated with the stroma of the cancer that are not found in the tissue surrounding it. And even further to that, we found that we have long-chain fatty acids that were localized in the stroma of the cancer, while the short-chain fatty acids were surrounding the stroma. So this points us to the direction that this technique is actually applicable to cancer, and there's something more important for us to investigate in that avenue. So we're still pursuing that. So this is something that's quite prevalent or relevant right now in South Africa. So I'm sure all of us heard about the news, and there's lots of cases of poisoning in the rural communities and in Soweto, and lots of children are unfortunately losing their lives. So in collaboration with Ziad and the CHAMPS network, we also got some liver samples from some of these post-mortem cases of children who died from suspected poisoning at Barra. So we ran 4 of these liver samples, and what was really interesting for us and kind of strange, in that 2 of these liver samples that we ran, we found the presence of naphthalene 1-diol. So this is a metabolite of naphthalene, which is common mothballs that you might use. So if you look at these images of the liver biopsy, the red color shows the naphthalene, and the green shows a structural lipid from the liver to show some contrast. In the 3rd liver biopsy that we ran, we were able to find the prevalence or the presence of aflatoxin B1. And this was around the same time where we had these challenges with, or we had this problem of peanut butter being recalled for aflatoxin poisoning. So maybe that's related to the case. And in the final example that I have, this nipecotic acid is a metabolite of an anti-seizure drug, a GABAergic drug, and we found really high presence of this metabolite in the liver of one of the children. So again, this is something that we're pursuing in the lab to try and investigate if mass spectrometry imaging can be a technique not only to identify the poison in the children that have passed away, but also try and understand what are the pathophysiological processes that we can then maybe treat or manage poisoning cases better in the future. So this is the last example that we have. So this is in collaboration with Bhavesh and Chris. So they are interested in looking at what causes TB bacteria to become resuscitated or stimulate them to grow. So they do this process where they grow the bacteria in vitro, they spin them down, and they can collect the cultural filtrate, which is kind of what's being secreted by the TB bacteria. So the initial test that we did was we took some TB bacteria, the cultures themselves, we put them on a glass slide, and we imaged them just to kind of see what they were different. And again, we looked at the total ion scan, and you can see from their lab strain compared to the MDR TB, we can see that there's really some very clear structural differences in these TB bacteria. We then went on to look at the CF, which is a cultural filtrate that's removed from these bacteria. Again, we can see that there's maybe not that many differences, or it's not as obvious as the TB culture, but it shows us that there's really a difference in what's being secreted by the bacteria and what's on the structural components. So it's something that we might be able to use to identify novel compounds for TB therapy. So this was also just a pilot, one example, something that we're still doing. So the advantage of mass spectrometry imaging is that there's some online databases that we can use to search for these compounds that we might find, compounds of interest. And just by inputting the data that we collected from this experiment, we found a list of maybe 300 metabolites that we were able to identify. And some of them are very specific to bacterial growth and bacterial function, like tRNA, tyrosine RNA, glucose-6-phosphate. So these give us an idea of how the bacteria is also growing and metabolizing, as well as some of the structural components. Okay, so like Aletta said, we really have quite an old mass spec, and Per is going to show you some of these really new fancy toys. So we have some limitations in what we can do, and some of our limitations are the acquisition time. So right now, Per is driving several Ferraris, and we are strolling around in our VW Beetle. The other challenge is that our instrument is limited in that we can do full scan MS right now, which limits some of our identification of compounds. And Aletta showed you the slide of how mass spectrometry imaging is really accelerating over time. So that means that the technology is also evolving at a rapid rate. And there's now MALDI-2. So we have a MALDI-1 instrument. It has a single laser. MALDI-2 has an initial first laser, and it has a second laser that strikes the plume that Aletta showed you in the first slide. We also then have really a big challenge with data and the space that we have. We find that we have to clean our hard drive on our acquisition computer and our data processing computer quite often because it fills up the data quite quickly. In addition to this, if you look at like a mass spectrum, there's 4,000 peaks every time we run an experiment. we look at maybe 200 of them, if that. So we touch a very small portion of the data. So thanks to Rob and some other collaborators, we're speaking to people about using machine learning and AI to try and make sense of this data and better understand what's going on. So we hope that through creating this data and creating awareness around the technique, we can start to address some of these challenges through collaborations and through bigger grant applications and more advanced instrumentation. So now I'll give the word to Pat.
Thank you very much, and thank you very much for inviting me today to give this presentation. I've been to South Africa a couple of times now, but it's always very nice to meet all the nice people here, be in this fantastic country, but also nice to escape the coldness in Sweden and the darkness. I will describe today what we are doing. As you see, I listed different MS imaging opportunities and also challenges, I would say, acquisition time. Sometimes it takes a very long time to run a tissue section. I will show you examples here of the primate tissue section that takes at least 24 hours with one single tissue section. Sensitivity is an issue. Identification could be an issue as well. Multi-omics is the next step where we try to do the same analysis in the same tissue section as spectrometry, spatial transcriptome, spatial proteins in the same tissue, and align the data to correlate spatial in each individual. Resolution is one of the questions I always get. What type of resolution can you provide me with? That's the most common question, actually, and data storage is starting to be a problem. We have 2 servers, I don't know how many terabytes we have. It's a continuous buying new servers because we're filling them up. In my lab, I have different types of mass spectrometers. You see 2 MALDI Fourier transform ion cyclone or resonance mass spectrometers that provides us with very high mass resolution data. So for each individual ion molecule we want, we can define it on the 5th decimal. And then we have 2 DESI instruments, and the most recent instrument is the MALDI 2 TOF called timsTOF Flex that Sooraj described. It has a MALDI 2 source that increases the sensitivity of certain compounds, but also it's very good because in that instrument you can trap ions and separate them. So if you have isomeric ions, you can separate them inside the mass spectrometer and get the identification, even if they have the same master charge ratio, same molecular. So we are trying to use these instruments to study the interplay of the neurochemicals and obviously trying to advance the understanding of disease and drug treatment. So one thing that we have been working on quite for a long time and very intensively is to increase the ionization efficiency of molecules that are very difficult to ionize. So unfortunately, neurotransmitters are one of those. So we synthesized this molecule, and there is one chromophore, as you see here to the left, that is aimed to absorb the energy from the laser. So that works as a matrix. The other chromophore is now targeting phenolic hydroxyl group or primaramines, and then we have a new molecule that flies very well in the mass spectrometry. So with this, we can now study the full catecholamine system, the full indolamine system, meaning that we can study L-DOPA, dopamine, norepinephrine, epinephrine, and all the downstream metabolites at the same time in one single tissue section. Same for 5-HT. And currently, we have identified approximately 100 different compounds. So in theory, we can do 100 different transmitters, metabolites, amino acids from one single tissue section. There are also other molecules in the tissue sections that are very difficult to ionize. That would be carboxyl acids and aldehydes. So we have published a couple of papers recently where we target those molecules. One target, for example, the TCA cycle, so we can measure all metabolites in the TCA cycle except one. And we also have recently published a paper where we can do imaging of cholesterol, which is pretty difficult to ionize, and hydroxycholesterol, which is a metabolite, and a number of different difficult to ionize compounds. I have two big projects in my laboratory. One is about Parkinson's disease and L-DOPA in dyskinesia, which I've been working on now for probably 30 years. Unfortunately, if I get diagnosed with Parkinson's, I have a few good years because I can take the replacement of the disappearing dopamine in the brain, L-DOPA. Unfortunately, about 10% of all the patients that are treated with L-DOPA will enter into a new phase called dyskinesia, which is kind of the opposite of Parkinson's. Excessive movements you cannot control. So that is very disabling for the patients. Very difficult to eat breakfast. Need a lot of care in order to be able to function. And it has a significant impact on quality of life. So now we're trying to understand why is I'm getting this, but not my neighbor who has Parkinson's. And after 5 years of treatment, about 40 to 50% of all the patients have this side effect. And you see this woman here having an on phase of Parkinson's, L-DOPA-induced dyskinesia. So using mass spec imaging, we try to measure as many different types of molecules as possible. So neurotransmitters, neuropeptides, metabolites, lipid glycans, but also transcriptomics and proteomics molecules. So we did this on a primate model. I'm very fortunate because I'm collaborating with a group in Bordeaux in France, and they have this biobank of tissue. So they sent me tissue because they wanted to do this studies. I think we have now like almost 800 tissue sections in the freezer. And I will focus on two groups. Both groups are Parkinsonian. They have exact same level of Parkinson's disease. This has been scored by trained people who know how to score Parkinson patients. It is not patients, but it's primates. And they have been treated for a long time, chronically treated with that. And one group developed these side effects, the other one not. They have received exactly the same dose. And then we annotated the different regions that we're interested in. And this image that we obtained a couple of years ago surprised me so much that I did not believe it. As you can see, this is the dyskinetic group, non-dyskinetic. Same treatment, same level of Parkinson's. Why is this group having 70 times more L-DOPA in the brain? But I can't answer that question today, but I will before I lay down. And what is very nice is that we can follow the metabolism of L-DOPA into norepinephrine, epinephrine, and all the downstream metabolites. So I told you, I did not believe it, so we repeated this 3 times. Now I believe it. Another thing we can do is to study co-localization of dopamine and serotonin, because there is a theory that L-DOPA being in the wrong place in the brain is taken up by the wrong neuron, meaning that L-DOPA is taken up by the serotonin neuron, converted to dopamine and released as dopamine in the wrong place of the brain. And this is the proof of that almost. So here is serotonin here's dopamine. That is not happening in the non-dyskinetic animals. And we can also follow different areas in the cortex where dopamine is not supposed to be in that high level as we see here. We have also analyzed neuropeptides, and we developed a method for that. So now we can image approximately 50 neuropeptides at the same time. We saw that the dyskinetic animals have increased levels of bioactive peptides. That was already known. But now we can also correlate this to, because we do this in the same animals, exact same animals. We can correlate the data we have from the neurotransmitters to the peptides, and we see that we have a correlation of the increase of neuropeptides in certain areas correlate to L-dopa and dopamine. And we also see that it seems that the brain is trying to metabolize the high levels of these bioactive peptides, because we see an abnormal metabolism of those bioactive peptides in certain regions. That was correlated to the severity of the dyskinesia. So now when we have behavioral scores, we have mass spec data, we can do these things. Another thing that was not reported before is that we also see most likely a change in the vascular system. So in the dyskinetic animals, it seems to be more vascularization, could be angiogenesis, indicating possibly increased blood flow there, or a leaky blood-brain barrier. Maybe that's why we have so much L-dopa. That's what we're going to investigate, hopefully together with Aletta and Sooraj. The second project is a bit different, but it's also about Parkinson's disease, but Parkinson's disease dementia and lewy body dementia. Again, if I'm being diagnosed with Parkinson's by my doctor, it's not really sure that I have Parkinson's. It could be something else that looks like Parkinson's, but it is not. And if I get L-dopa as the common treatment, I feel very bad. There's very few, or there is no biomarkers actually for diagnosis to differentiate between Parkinson's disease and the so-called atypical Parkinson's symptoms. You can do that when the patient has died and you have the brain. So what this project is relatively large. It's a research environment grant. It consists of 6 groups, from the organic chemist to the neurologist. And in between, we have the different spatial omics technology, spatial mass specs, spatial transcriptomics, spatial proteomics, obviously bioinformatics. And this is what we're working on right now, because we have post-mortem brain tissue, we have cerebrospinal fluid, we have plasma samples. So what Sooraj said was that we can now take 1 microliter cerebrospinal fluid, completely untreated, directly, if you like, from the patient, put it on the multi-target plate, and we then apply this FMP10 matrix that targets neurotransmitters. We then analyze with our mass spectrometer, and we can identify directly from that 1 microliter, approximately more than 30 neurotransmitters metabolites, and we can separate. So this was a pilot study. We can separate de novo Parkinson's patients, meaning that these patients never received any treatment, from controls. So obviously we are now repeating this in a much larger cohort, hundreds and hundreds of samples, but it looks very promising. Another thing that we're working on is to do this different types of analysis in the same tissue section. So in this case, we are doing lipids, both in negative mode, positive mode. Then we wash off the matrix. Then we apply an enzyme, so we release the glycans, we measure the glycans, and then we do a wash again of the tissue section, and then we do a trypsination to measure gyptic peptides, proteins. This is unpublished. And then we can create these images. This is again a Parkinson's disease model. We have 3 different glycans areas to show you how it would look like. Another thing that we are working very, very intensively on right now is to integrate different spatial modalities, such as spatial transcriptomics. This was a paper we published last year in Nature Biotechnology, a year ago approximately, where we can take different matrices, neurotransmitters, metabolites, positive ionized metabolites, negatively ionized metabolized, and we take the same tissue section and do then spatial transcriptomics, correlating the data, which actually looked very, very nice. I don't have time to go into the details of the results. As I said, one of the questions I'm always getting, what resolution can we do? And this is something that many people are working on, this spatial or lateral resolution of mass spectrometry imaging. And there has been a number of different approaches, laser beam shaping, so in publication that there is a commercial source actually that can do 1.4 micrometer sized pixel or laser shot. You can improve the ionization process. You can, as we do, create new matrices. You can do oversampling, as you see down there, or even sample stretching. Someone put the tissue on a paraffin and then stretched it and analyzed it, and then put the data together. And there are different imaging co-registrations and image fusion methods. But these methods can obviously improve image resolution, but there are also fundamental drawbacks because they come from different sources. And what we are also fighting right now is to align tissue section that comes from different modalities. That's not easy. So what we tried, and this is also very preliminary, so it's not published, but we are using a deep learning method approach. We are calling it deep convolutional neural networks with super resolution enhancement methodology. So what we did was we acquired an image at 50 micrometer resolution. We downsampled that to 150. So we created a bad image, so to say. And then we trained this system to improve the resolution until we reached. So we go from this one to this one that's supposed to be the same as that one. So we reconstructed that low resolution to the high resolution image using this deep learning method. And then we analyzed or imaged. This is an example of hippocampus from 3 different regions. As you see, at 5 micrometer, magnification looks very fuzzy. And then we put this data into the deep learning algorithm. And you can see the improvement in resolution. This is probably individual cells in the hippocampus. We then also looked for neurotransmitters. This is in GABA, where we did the same. We ran at 5 micrometer lateral resolution, processed the data, and this looks very, very good, actually. I do believe this, even though it's like magic for me, because GABA cells does not have any GABA in this nuclei. And then we took the consecutive section to verify that it is actually GABA neurons, and we count to stay in the same section. We dop it to visualize the cell nuclei and overlay them. So it looks good, but it's very, very preliminary. So what is the future of mass spectrometry imaging? What is next? I think improved resolution by using AI or deep learning method is coming very, very soon. And instrumentation is going to be better and better. The timsTOF Flex that we are using is taking 15 pixels per second. That's going to increase. I mean, actually the problem is to transfer the data from the mass spec to the computer. There is a limitation there, and that is a problem right now for the mass spec companies. To integrate with multimodal analysis, as I said, we are working heavily on this, and we are not the only ones that are doing that. Sensitivity is always an issue, and the technique is moving more and more into clinical applications, that's for sure. And broadening molecular scope, yes, we are developing different types of chemical tools to analyze or image as many different diverse molecules as possible. But one thing I think is really, really lacking, and that is the standardized protocols between labs to compare data. By that, I would like to thank you. Here you see part of my research group. We were here last year in November, of course, and had a workshop here in Johannesburg. So thank you for listening.
On a more philosophical basis, you know, what you were presenting, I perceive as looking at smaller pieces of the puzzle. But isn't the picture still important? So it doesn't matter how small the pieces are, you know, if you put the pieces together, then it's still the same picture.
Yes, but sometimes I think the picture needs to be looked at with a microscope in order to find, so for example, correlate transcriptomics data with proteomics data with mass spec data could give us more information if it's imaged. Was that a good answer to your question or not?
So I think my question's a different take on the philosophy of the research. So it's the internal logic is absolutely clear and fascinating, and thank you, all three of you, for a great presentation. How do you, who's involved in defining the research questions? How multidisciplinary is that team? How do we know what to ask or what to look for? I'm really curious to know about that process.
Right. I can only answer for myself, but I'm, for example, for Parkinson's disease, I work very closely with the neurologist. He sees his own patient, and we get samples from him, both plasma, CSF. He's also professor of King's College in London that have a very big biobank of human post-mortem brain tissue. So absolutely we are trying to work with the best persons that can help us with asking the right questions. Yes. Absolutely. And we do, but
Institute in South Africa that is interested in understanding mediators of cancer. Could you, and you're looking for pathogens or microbiota or something from microbes that has somehow stimulated the onset of cancer. Could this technique be used for that? If you had the tissue, and how would it be used?
Absolutely. This is what many different research groups are doing right now. We have not yet been so much involved in cancer because that's probably because of my own background in neurodegenerative diseases, but we are collaborating through the SciLab Lab platform with different persons who do cancers. We do prostate cancer, we do breast cancer samples.
Yeah.
What else do we do? There is a
But let's say a virus is responsible for stimulating prostate cancer, for facilitating prostate cancer. Do you think you could find the signature of that virus in tissue if it's still there? If it's a hit and run from 20 years ago, I assume not. But if there's still a presence, and how would that work? Are there characteristic signatures of each microbe?
Question. I don't think I pass on that.
Yeah. Well, I mean, I think, Sooraj, you may have some thoughts.
Yeah, so I think to go back to the research question, I think the research question is very much directed by the researcher, because I can give Lizette for an example. Lizette came to us with her mosquito eggs and some mosquitoes, and one of the challenges they have is looking at the diagnostics of the mosquitoes and if they are malaria-carrying vectors. And normally it's done with electron microscopy, but using mass spec, for example, we can create a fingerprint or lipid profile of the surface of the mosquito egg. And each species should have a unique profile that predisposes them to becoming a malaria-carrying vector. So this is how we kind of, when people come to Aletta and I, we try to have a discussion around what are some of the challenges and how we can tailor the technique to them. Because I think the advantage of the technique is that in theory, everything or every biomolecule that's relevant has a molecular weight. And if it has a molecular weight, we should be able to measure it with the mass spec. So that's the kind of thinking that we approach it with. And I think like with your question, Rob, and it's kind of similar to what we see with the samples that we got from Baijnath, where we took the cell culture media and we took the bacteria themselves. So there's differential biomolecules in each of these, and we can then look in the cancer if it's present, if it's present in the circulation, and then try to create these profiles. And this is something that we talk about a lot now, is that we don't have the computing power. Like we said earlier, there's 5,000 peaks that we create. So somewhere in there is a unique fingerprint for each of these cancers or cancer-causing viruses that we are not able to see because there's tons of data that the human mind cannot distinguish. So I think by, like for example, the Mind Institute, we can leverage those kind of resources. Then we can create a database of prostate cancer viral particles and be able to compare them, look at which of these biomolecules are driving the process.
And I also think that in the future, we will be much better in identifying peaks in the mass spectra, because that's one of our main problems. So with the FTICR technology, we see thousands and thousands of peaks, but they're very, very difficult to identify, because if you go to a database of a certain mass to charge ratio, you will see maybe 25 lipids there. Which one is it? And that's a challenge that I think we're facing right now. But if we can solve that problem, we would write a science paper every week. Yes, it's coming. It's coming.
I just want to add to that. So in terms of when we started this, we really said, to answer your question, that we will work with the individual researcher in order to define the question. So we obviously do not know every field, the relevant questions. And that's really what we're trying to do, is have a robust conversation. What is it that you are interested in? And then coming from the technology side to say, what exactly is it that we can help in order to answer? So we are not driving all the research questions, definitely by no means. We try and partner with the research groups and the individuals in the faculty, and a lot of people have approached us, and I think there's actually quite a lot of interest and excitement around this, and this is why the seminar was brought to fruition, so that the technology is available for people in the faculty, and that's really what we want to do.
Minutia level, I think there are certain, I think you alluded to this, there are certain molecular weight compounds that aren't ionizable, I think. And do you have a way to supplement, to sort of fill that gap if those happen to be important with other techniques?
Sometimes we use a different ionization technique called DESI, desorption electrospray ionization, that is very complementary to MALDI that we talked about today. That's one. Another way is to create those specifically designed matrices for specific types of molecules that we are working on.
I think right now with MALDI, we have so many matrices available that we we actually have quite a wide coverage of analytes. I'm not sure if we had to guess the currently available matrices in 2025. So about 25 matrices, and each one gives you access to maybe 2,000 each on average. Being very conservative, so we have access to 2,000 biomolecules that we can detect.
No shortage of biomolecules. Yeah. Is there any way you can imagine doing this sort of detection in vivo? So I mean, for the Parkinson's work that you described, Per,
Yeah.
you got to get tissue to be able to know, where the patient.
There are
falls.
companies now that are developing at least 3, I think, that is developing the DESI technology. So, for example, if you operate a intestine cancer, the surgeon wants to know when should I stop cutting, right? And this pen is helping him when sometimes they cut with laser, so that creates fumes. And that fume contains characteristic molecules from cancer tissue or healthy tissue. So the instrument already have a database of different types of tissues. So the surgeon looks at the screen, and it's green when he's going to continue cutting, and red when he should stop, based on the molecular profile from that cancerous tissue. Yeah.
That's right. I mean, is that not cool? I mean, part of why we wanted to do this seminar, and thank you, all 3 of you, for really wonderful presentations, is to raise consciousness about this capacity. Clearly, we have to also raise consciousness with donors and also funding agencies, so that we can fully have state-of-the-art capacities here. But you've done a lot with, what would you say, previous generation, I guess,
Also, at least
10 years
generations
back.
old.
Yeah. And 10 years goes by so quickly.
Especially in aspects.
To the question that you asked is what are our priorities, and how can we move this forward and seek funding to get to a point that we can be first in class in Africa? I think you are already first in class in Africa, but maybe we're could go, given the questions that we're trying to answer here, I think, as you said at the outset, are different than what people might be looking at in the North. I think it could be globally first in class if we have this capability. This has been a great seminar. Thank you all 3 very much.